This tutorial is about how to carry a basic genetic diversity analysis. In a genetic diversity analysis, we study the variation among and between individuals, groups of individuals, or population. You could ask yourself, why is it important to study the genetic diversity of organisms? First, because it constitutes a useful support for crop improvement strategies, as it allows, for example, for the identification of potential parents and parental combination for breeding programs. Second, it helps the conservation effort of valuable genetic resources by helping to focus the efforts on the population that need it the most. Genetic diversity can be analyzed at the different levels: diversity of individual, population, or species. This is carried out using a variety of data, such as pedigree data, not suitable in natural population, as this information is hardly available. Also biochemical markers are isozymes, which are no longer used for this purpose, and molecular markers evaluated by PCR amplifications, such as AFLPs, RIPDs, and microsatellites. These markers are very commonly used, but are being abandoned in favor of single nucleotide polymorphisms, or SNPs, that are very common on the genomes and are now very easy to evaluate thanks to the next generation sequencing techniques. In this tutorial, we are going to analyze data from a Coffea canifera germplant collection conserved by the Cirad in the French Guiana. Coffea canifera is a rubiaceous plant originated from the sub-equatorial plains of Africa. It is used to produce the Robusta coffee. The SNP data was obtained by the sequencing-based diversity array technology, or DART-seq. Part of the data here analyzed has been recently published in this paper from 2016. Our data is organized and coded with individuals on columns and the markers on rows. The reference nucleotide is followed by the SNP nucleotide separated with a slash. We're going to use R and RStudio. As we did, we have the possibility to follow and execute the script simultaneously using an R Markdown file. Finally, we're going to use the R packages AdGenet, exploratory analysis of genetic and genomic data, that we are going to use to carry multivariate analysis of the SNP data. We're going to use Popper package, used for genetic analysis of populations with mixed reproduction, in order to calculate genetic distances and create trees with bootstrap support, and the ape packages for analysis of phylogenetics and evolution that we're going to use for reading and manipulate phylogenetic trees. What we're going to do is perform a discriminant analysis of principal components, or DAPC, which is a multivariate method designed to identify and to describe clusters of genetically related individuals. With it, we're going to identify the number of groups and group memberships, visually assess the population structure. We're going to classify unknown individuals, and we're going to obtain and visualize the, the membership probabilities for each individual. And finally, we're going to obtain a neighbor joining tree supported by bootstraps. So let's start with the analysis. First, you have to have R and RStudio in your system. You have to have also the packages that we're going to use, Popper and AdGenet, installed. If you don't have them, you could use the install packages function of R. Then you have to load the libraries like this. Okay, they're loaded. And we have to set our working directory, the directory where everything is going to be stored. We're going to run this command. Okay. Then we have to import the data to R. To do that, we're going to use the read delimited function. We're going to give it the name of the data file. We're going to tell them that it doesn't have to import the header, or the file doesn't have headers, and that the data is going to be imported as it is. So we need the names of the columns will be in the first row, and the name of the rows are going to be in the first column. So we're going to tell them that, and then eliminate those columns from the data. We have to do that because if we import the data with the header names, sometimes the characters are not accepted by R and it changed the name, so you could have problems with the data. So let's run this. Okay, it's done. And now to check the file that we have. So this is our data, how it is imported. We have, in this case, we have the individuals in columns and the markers in rows. And we have the first letter is the reference letter, the reference nucleotide, and the second letter is the SNP. So, okay. Okay. Then we need to import the data, the object data, to the package that we're going to use. And for that, we're going to use the df2genin command. We're going to tell them to take the data that we already have imported here. We're going to transpose this file because the software expects that the columns represent the markers and the rows represent the individuals. So we have to change this in this case. We're going to tell the program that the ploidy of the data is 2, that the separator that we use to separate the alleles is slash, that we have codominant data, and that we are missing data character. It's this one. So let's run this. It will take a while depending on the name and the number of data that you have. Okay. It's important that you know that you could also import data from other formats. You could check this vignette where you could find information on how to do this. Okay, it's done. Okay, so you suppose that your data have two sets, different sets of individuals. Ones that you already know the groupings, already have classified them, so you're going to use them as a reference. Others that are unknown. So we're going to subset our individuals that way. First, we have a list of the names of the individuals that we know, and we're going to import this. And then we're going to subset the genin object that we created to get only the data of these known individuals. Okay, so let's check the file that we created, the object genin known. Excuse me. Let's check it. So we have this object with 113 individuals, 2,719 loci. We have the table where the data is stored. We have other information here, like the number of alleles, the list of alleles name, the ploidy, the kind of data, and the call that we use to create this object. Okay. Now, if you have the information about the groups to which each individual belongs, you can import it here. If not, we have to get this information, obtain this information, and for this we could use the find clusters command. Let's run it. Okay, now the command is asking me how many principal components are we going to retain. We're going to retain most of the variants possible, so we'll say 120. Okay, and now the program is asking me how many clusters. there are. I will check and we'll say around 7. About from 7, it's okay. 6, 7. Okay, let's say 7 clusters. Ready. So what do we have now? We have a object called groups that stores the information about the groups that the data is organized. We check this. So we have each individual here and the group, the common found for each one. Now we are going to run the discriminant analysis of principal components. We're going to do it by running this command and passing to it the genInt object that we created with the known individuals, the groups that we found for each one of them. Let's run it. Okay, the program is asking me again how many principal components am I going to keep. This is very important because if you keep too much information, you overfit the model, and if you don't keep enough, it won't be explanatory enough. So if you're not sure of how many principal components you're going to keep, you could run these two commands, this one and this one. That could give you an idea of how many principal components will do a good result. So I'm not going to run them right now because it takes a long time. I already know that my past numbers is 6. Okay, so once again, the program is asking me how many discriminant analysis agent values or discriminant functions am I going to keep. I could keep 5 or 6 or 4. I would keep 4. Okay. So we have our analysis already. Now you have the data, but you have to plot it. You have to visualize it. We could do for this a scatter plot. I'm going to create first a vector with the color names, with colors. You could check or you could use the default colors, but sometimes they're not very nice. So I'll create this one. Okay, so I have my vector. Now I'm going to create my scatter plot with the first and second discriminant x. Let's run it. Okay, so I have my plot with the 7 groups that I found here, here, here. Okay. I'm going to put the axis to the plot. Okay. So you can easily export these plots with this function here in RStudio. Then I'm going to do the same with the other discriminant axis that we have found. Okay, this one. And once again, we're going to put the axis on the plot. Perfect. So we have here a plot where we could visualize results of our analysis. We could find distribution of our groups. Okay, now we have our control individuals. But what about the ones that we don't know? We don't know which group they belong. We could use the predict function from the program, from the package, to identify these individuals. First, we're going to import the list of names of the individuals that we don't know. I'm going unknown. Then I'm going to subset the genInt object with these individuals. And last, I'm going to predict the individuals within this space, with this scatter plot. Okay, so let's run it. Done. Okay. And we're going to create the scatter plot for this. Okay, let's run it all together. Okay. Here we created a scatter plot for the first discriminant x, and then I plotted the unknown individuals on the both axes. So this is our scatter plot with the unknown individuals, where they're located. And again, for the last two discriminant functions, okay, perfect. Once again, if you want, you can export the image here. You create a bar plot with the membership probability of each one of the individuals analyzed. You could use the complete plot common of the package. Here I'm going to use a bar plot common because I want to plot together the known and the unknown individuals. So let's run. Okay, here you can check the membership probabilities for each one of the individuals. This is more or less like a structural plot where you could see how well each individual belongs to each group. You can easily detect any admixtures that you have within your individuals. Okay. Finally, if you want to create a tree with all the individuals, it is possible running this function from the popular package. So here we're going to do a neighbor joining tree. Then we're going to plot this. Okay, let's do the tree. It will take a while, but it calculates the data. The plot tree. Okay, it's done. It gives me a tree, a file with a tree in it. You could plot it with colors because it's not very clear like this. Okay, that's better with colors. Okay, and we can plot also the bootstrap for each one of the branches. Okay, perfect. This tree can be exported and you can open it after there on any other software to visualize this tree like Fig Tree. If you have doubts or need more information about R, you can go to the CRAN site, and from then you can access to an infinity of resources to learn to use the language. For help with the packages, you can check the vignette, manuals, and documentation of the web, or directly on R by typing the command help and the name of the package. For more information on genetic diversity analysis, you could go to this website that has a very comprehensive and explanatory explanation of how to conduct different genetic analysis. This video tutorial has been possible thanks to Suntory. This tutorial is about how to carry a basic genetic diversity analysis.